Welcome to this poster presentation on InParanoiDB 9, a website for exploring a database of ortholog predictions on the level of full-length protein sequences and protein domains for 640 species. Orthologs, commonly defined as genes with a common ancestry separated by a speciation event, tend to share function across species and are therefore highly useful in bioinformatics. Orthologs can, for example, be used to transfer information on drug targets in model organisms to human, or to transfer evidence between species in functional association networks. Orthologs are commonly inferred on full-length protein sequences, disregarding the fact that proteins can consist of domains that can have different evolutionary histories. This type of discordant domain orthology requires domain-aware approaches to be found, and when inferred, they can give a more comprehensive and fine-grained view of the evolutionary histories of genomes. The InParanoid database is a well-known resource for ortholog predictions with over 9,000 users in 2022, and its underlying algorithm has been seen to perform well in benchmarks. However, the database was last updated in 2013, making many of the underlying proteomes old and outdated. The InParanoiDB 8 was built on the InParanoid algorithm and does therefore not include any domain-aware orthology. We therefore created InParanoiDB 9, an update to the InParanoid database, including orthologs for both full-length protein sequences and protein domains. We used the InParanoid diamond algorithm to obtain full-length protein orthologs and domainoid to predict orthologous domains. A new database and website was built to allow for simple navigation between full-length protein and domain orthologs. The resulting InParanoiDB 9 includes 640 species and a total of over 1 billion ortholog groups. Inclusion of full-length protein orthologs as well as orthologous domains allows for exploration of orthologs that would not have been detected using traditional orthology inference databases. Now, we're going to take a look at an example on the InParanoiDB 9 website. So let's search for this protein P22102, or the GART protein in human. Let's say that we are interested in finding orthologs in E. coli. So let's input E. coli here in the filter. Here we can see that there are no full-length protein orthologs between the human GART protein and E. coli. We can then instead click on one of the domains on the GART protein in human, for example this GARS-N domains, to see if the domain has any domain orthologs in E. coli. And we'll filter again for E. coli. Here we can see that there is an ortholog group for the domains GARS-N in the human GART protein and the E. coli pure D protein. Let's click on this and view it in further detail. Here we can see the detailed view for this ortholog group. We can see a tree from the sequences built from the orthologous domains, and we can see a drawing of the domain architecture in the human protein and in the E. coli protein. Here we can see that these two GARS-N domains are highlighted as they are the ones that are orthologous in this case. In this table down here, we can see some additional information on the orthologous domains. And in these tables in the bottom, we can see orthologs for all of the other domains in these two proteins. So by utilizing this function, we can, for example, click on the next domain in the human protein, the GARS-A domain. We can then see that this GARS-A domain also has an ortholog on the E. coli pure D protein. The same is true for the next one, the GARS-C domain. However, the next domain on the human protein, the AIRS domain, is orthologous to a domain on a different E. coli protein. We can view it in further detail by clicking here. Here we can see a drawing of this E. coli pure M protein, which contains the AIRS domain. We can also see that the next domain on the human protein, the AIRS-C domain, is also orthologous to a domain on the pure M E. coli protein. Then we can also see that the last domain on the human protein, the formal trans-N domain, has an ortholog on yet another E. coli protein, the pure N protein. In UniProt, the human protein GART is described as a trifunctional enzyme catalyzing 3 different reactions annotated with the same 3 pathways and catalytic activities as each of the 3 corresponding E. coli proteins. This is an excellent example of domain orthologs in InParanoiDB 9 that would not have been detected using a database of orthologs predicted on the full protein sequence level. Thank you very much for listening. If you want to know more about InParanoiDB 9 or if you have any questions, please send me an email or come visit my poster.